I created a separate link, and it was my own fault.
All
I'm
right.
shot myself in the foot. How are y'all doing today?
Pretty good.
Good morning.
We're having a little computer difficulty here. I won't be able to pause or hit. I can't shut the recording off, so just heads up on that. We will try to edit out anything if we need to. So,
Okay.
but we got looks like a pretty good crowd on here, Josh. If you want to take you and Kennan want to take it away, RhizeBio update call. So,
All right. Well, thanks again. Appreciate y'all for working with us this year. Just to kind of set the stage for what I think a good agenda for today might be. We have these report outputs, as I'm sure you're aware from last year, that we're going to go through as a kind of an example template of what you could expect. And then also, we have a new and improved updated soil and rhizosphere root report. And then kind of closer to the end of the meeting, I'd like to get y'all's thoughts or opinions on, we're looking at launching a new kind of direct to farmer program that integrates and implements some of these cool technologies for being able to make decisions on seed selection and biological inputs. And so, Kennan, I know that you've been we've been working on these reports for the ag launch trials. We have them all completed. We're just adding in our interpretations into the report. So a little different from last year. You're not just going to have the report with the different biomarkers that we're looking at. We also include key findings and then a summary or interpretation of what that data means as it relates to your farm and maybe areas that you could look for improvements. Last year, I think a key difference between this year and last year is that we were trialing different biological inputs. So input manufacturers would come in and they would pay for and or sponsor a treatment on your farm. And so we would basically run their product in furrow, typically inoculated to the seed, in order to look at the rhizosphere in a greenhouse setting and inform on whether or not that inoculant would perform. This year, we're doing things a little bit more differently in that we're just looking at your soil and your seed combination and the grow out in the greenhouse. So, and we feel as if we've seen enough of this data in order to indicate areas of deficiency. We've seen the way that these different inputs and stimulants work that we can provide some feedback when we go into our one-on-one meetings after we deliver the report. That's going to provide a high level of value. So that's kind of what we'd like to discuss today. It would be really helpful if y'all could think it through as we're going through some of this data, ways that you might recommend us to maybe cut back on information or improve the way that we display the reports would be really helpful for Kennan and I as we're going through this new iteration of testing data. So without any further ado, I'm going to hand it over to you, Kennan, to kind of go through maybe an example report that we've produced for this new batch.
Yeah, I'll go ahead and share my screen. Like Josh mentioned, this year, or as of right now, we're just kind of looking at single reports, and we're looking a little bit more at kind of how the stress response basically that plants have and the microbiome's ability to help the plant respond to stress, and how that relates to how a biological is going to be able to integrate, what kind of role it's going to be able to perform. And so I'll kind of run through the report, I think. I see some familiar faces and names on the call, so I'm sure some people will be a bit familiar with what we're measuring, how we're measuring it, but I'll give just kind of a baseline to get everybody up to speed on what we're measuring and how we measure it. And then maybe I'll talk a little bit through a specific result just to kind of show how we use the data. And then, like Josh mentioned, we are going to try this year to incorporate some kind of interpretation, because we know that these reports can end up looking like just big walls of numbers, and just to kind of help farmers real time be able to learn how to read these and understand them a little easier without us having to not that we mind jumping on calls, we like jumping on calls, but kind of help out with that. So I'll go ahead and share my screen. So I'll bring up a couple reports here. These are all in-network farmers that participated this year. And one of the interesting things is we sort of asked most of these farmers to provide, if possible, like kind of a lower performing area of the farm and a higher performing area of the farm that helps us contrast where things are working well and where things are not, or how the microbiome is kind of responding in those problem areas to try and still help the crop do well, even if it's not, the best soil, or is an area that's been, kind of farmer just bought the field, it's been treated poorly for a number of years, things like that. And so we will see, say a difference between a farm that a same farmer's been managing for a long time versus kind of a newer area. And often we will see differences, really significant differences. So I'm going to highlight some of those. First and foremost in the report, we look at the kind of community structure. And so we're looking at the number of genera, that's basically the number of different types of microbes we see. And we look at two different ways of measuring basically how diverse and competitive that those microbes are against each other. And we like to see those numbers nice and high. That usually means that there's a lot of different roles that can be filled, different microbes that are able to perform different functions when they're needed, even if they're not needed all the time. We always look at mycorrhizae, always a really important one. This was an interesting field because often when you don't have very good evenness, there's not a good chance for mycorrhizae to really get out there. But this field saw really high mycorrhizal populations relative to some of the kind of other facets of the field. We always look at fungal to bacterial ratio. This field was actually, we would call this a little bit fungal. About 1 to 3 is balanced on our report, and this will be, we'll kind of add some notes like this to help people interpret their reports when we send them out. And this also is very ectomycorrhizal versus endo. So we're seeing a lot more breakdown of nutrients reaching out into the soil and breaking down nutrients than we are delivering those nutrients to the crop roots. We always look at oxygen environment. We're really interested in how the microbes are getting oxygen, because that's going to kind of determine downstream what microbes are going to be able to survive well, and what roles they're going to be able to play is based on how much oxygen is available. We always look at carbon. Obviously, carbon is very important. This is a good field, a good example of really high carbon fixation. We like to see that. We're drawing carbon down. This is going to add to those kind of carbon sequestration scores, any of that sort of stuff, and just building your soil OM profile over time. And we also have good carbon breakdown. And so this is going to be taking, corn stubble, any kind of litter that's on the soil, or any manure or compost that you've applied. You're going to be breaking down those carbon sources into something that the plant can actually use. So this is also a good metric. And we keep methanogenesis down. And this is basically a loss of carbon, right? We're going to turn that carbon back into gas. We just grabbed it out of the atmosphere as gas during fixation, and we're going to turn it back into gas and lose it again. So this is a really great carbon score, and a field that I know was kind of a problem spot for this farmer, but we can see that at least he is making some gains as far as he has mycorrhizae, and we are going to be adding carbon to that system, which is going to year on year be able to show us some improvements in how the microbiome is going to be able to respond. For nitrogen cycling metrics, obviously nitrogen is kind of our most important primary nutrient other than carbon. This is an interesting field because we didn't have a whole lot of N-fixation. I think it probably just due to the time of year, there was nothing planted yet. And yeah, we would like to see that N-fixation number go a little bit higher for soybean, but we're seeing really good breakdown. And again, that's kind of similar to carbon, that there's a lot of nitrogen in different inputs. And how well are we breaking that down into, ammonium or urea or nitrates, or usable forms of nitrogen that the plant can actually take up. And so this system is actually getting most of its nitrogen from some sort of organic matter input rather than from N-fixation. And so that can actually kind of tell us a lot about how the system will respond to change and how it's going to deliver nutrients to the crop effectively. Phosphorus and potassium solubilization, obviously very important, especially, I guess, a little bit less in this area of the country where we're at. Down south, phosphorus gets really, really bound up in the kind of iron-rich soils, but phosphorus can get bound up for a lot of reasons. So we find this marker to be very indicative of the plant's ability to kind of get phosphorus out of the soil environment. So 50%, right down the middle. Just to add that little bit of clarification, because I think I kind of skipped over. These are all going to be percentile rankings. So what this means is we took this soil, we counted up all the genes we could find, and we compared how many we got against a reference population of all the other similar soils that we tested. So in this case, for soybean, there was soybean in this field. And so this means that we had basically perfectly average phosphorus mobilization for the soybean fields we've seen, but very high carbon fixation compared to other soybeans and really low nitrification and denitrification. So good nodulating bacteria, and we will be able to see that in our species count. We did have a reasonable population of Grady rhizobium and mesorhizobium. Could be a little higher, but we did have a good amount of nodulators that we found. Sulfur reduction and oxidation, these are a little bit low and a little bit pointing towards reduction. I always like to look for that. I want to see a little bit more oxidation and reduction. This was a really interesting result that I'd like to bring up is calcium transport. Often we see changes in calcium transport related to liming. If the farmers had to lime their field for pH for any reason, we'll often see a big reduction in those calcium transport genes because there's so much calcium just in the soil ready to be grabbed. Iron acquisition, this soil is doing a really good job of scavenging for iron. Not as important as some other nutrients, but still can be. And so we always like to include some of these, they're called siderophores, and they can also help with other kind of trace minerals and stuff, manganese, magnesium, things like that. One of the most important markers we always like to highlight is our plant stress adaptation. This is going to be what a lot of people think of when they think of what the soil microbiome can do. This is auxins and gibberellins, different phytohormones that can actually help the plant grow directly or help it deal with drought stress or pest pressure, things like that. And so 69 is a pretty nice high score here. I always caveat this that, I kind of want to see a balance there. About 70 to 80% seems about ideal. If you're hitting 99% on stress, that might be a bad thing, right? You have lots of genes that are working hard, but maybe your system is a little stressed. You have a drought, or it's been waterlogged, or pest pressure, something that's driving that marker up. So I know I kind of just threw a lot of information out, but this is sort of the report, and that's a good example of why we're going to try to integrate these sort of key findings that we've been able to develop that just look for some of the key indicators that I've mentioned about, rhizobium populations and kind of an understanding of nitrogen cycling. Is it front end loaded with a lot of fixation, or is it back end loaded where you have a lot of nitrates and denitrification happening? Looking at the mycorrhizae, different things like that. So we are hoping that this report, I think, will be able to provide both kind of a baseline understanding of what the conditions are on the field, as well as, like Justin had mentioned, the ability for the soil to respond to stress. Because at the end of the day, we think that's one of the big things that the microbiome is able to perform is to make sure that the plant isn't taking big losses, even when there is, weather issues or chemistry issues or pest issues or whatever's going on. The soil will kind of help to fill in those gaps. And so this is one thing I would point out here. We saw a really high population of a couple of bacteria, and that's exactly why we had really low diversity and evenness. So this would be a system that would be a great candidate for something like a complex biological amendment, where you're actually introducing new species, you're kind of improving the competitiveness of the system so that some of these kind of lower numbered organisms can start to compete with this Enterobacter and this Priestia, and that will sort of unlock some of these pathways that are a little bit crowded out, a little bit shouldered out in the soil right now. And we do boil all that down into a Rhize score. It's on a 0 to 10 scale, but effectively it's usually between about 3 and 6, 3.5 and 6. We're still holding on, waiting that someone's going to come across with a 9 or something, but that doesn't seem very realistic. So, and I was going to run through just some of the kind of differences that we can see, just how different two different fields can be. And so I guess these are different states, very different soils, but we have about 600 more genera, and some of that is due to the management styles and different things like that, and some of that's just going to be due to the local soil. Some of that's to be due to how much clay and how much silt and how much organic matter you have and what the organisms are able to do. Interestingly, we see a lot higher diversity on this sample, but we actually have a lower mycorrhizal population overall. And I would kind of think that the other organisms, the bacteria that are in these, we have a lot of healthy Pseudomonas, we have really high Brady rhizobium, so we're getting a lot of good end fixers in there as well. Um, are performing some of those functions, so the mycorrhizae are not quite as high. But as you can see, this has kind of a more balanced profile, right? We're not seeing really high scores or really low scores, but it's still looking good, right? We're still having more nitrogen breakdown and fixation than we are nitrification. We're still having good carbon fixation and breakdown and less methanogenesis, even if it's not so heavily kind of sequestering. This was a very much a sequestering carbon system. We're adding a ton of carbon to this system. This one, not so much. It's kind of more balanced, more in stasis. Again, we actually have slightly lower phosphorus and potassium mobilization, but this is one thing that I always note is if you have a very kind of a, what people tend to call like a spoon-fed program, a very prescriptive program, you're adding nutrients or foliars throughout the year, oftentimes we'll see some of those kind of nutrient pathways slow down a little bit just because they don't need to be that active. The nutrients are there when the plant needs it. And we actually see, I guess, a similar stress adaptation score, but the point is that all boils into a little bit higher rise score. It kind of does value that balance and having all the different roles occupied so that the microbiome can be really adaptive to different situations. Um, and again, yeah, a little bit more balanced. And we do always do a pathogen screen as well. I should have mentioned that we have added some nematodes to our pathogen screen. So we think that'll be a very helpful thing for helping farmers understand where some kind of management decisions might be able to come out and comparing different fields to understand what decisions they've made in the past, what effects those have had. If you have been able to reduce, root knot nematodes or fusarium wilt or something like that in the field over time. And so, yeah, I think that's kind of the basics of the report. And then, yeah, like Josh mentioned, we are going to be rolling out our kind of interpretation to help farmers understand a little easier without having to have me or Josh come talk their ear off for a while about what we think their soil's doing. So, Josh, do you have anything to add on the kind of report itself? I know that was a mouthful.
Yeah, couple of things that we're considering. As y'all know, we do a lot of work with different input manufacturers, and so understanding what those companies' products are doing, that's really kind of the unique part of our technology is telling telling the farmer, hey, this is the mode of action for this product. If we pull a report and we see that you have really high nitrogen fixation and you have a company that's saying, hey, you should buy this product because it has a lot of nitrogen fixers in the product. It might justify not making that purchase because you already have pretty healthy levels of nitrogen fixation or whatever it may be. And so in the future, we I guess as a company, our position is we are an unbiased, transparent steward of information. We don't necessarily like to make recommendations just from the machine learning models that we've developed, but putting in a section that might say, here are a few considerations of, hey, you might want to add a humic or a fulvic or some sort of compost extract in order to we know that those products have these typical functions, and so it might be that these products have a higher likelihood of integrating into that system. So those are things that we're looking at and helping to provide insight into into how to interpret this data. The other thing is if you're looking at different seeds as well, we're learning that different seed genetics can impact the way that these biologicals are performing. Some seed genetics might discourage certain fungal populations or certain abilities in the soil or maybe it improves, ectomycorrhizal populations. those are things to consider as well whenever you're making decisions on not just seed genetics, but also biostimulants or biofertilizers that could potentially be a good solution for you. So I know it's a lot of data, but this data is a means to helping to you to make the right decision out in the field. And I think it would be good maybe to jump in. I can share my screen with our, I guess, new and approved,
Any
updated.
any quick questions before you want to jump into that, Josh?
Yep.
I got a few, but I figured I'd yield to everybody else first. So feel free to unmute yourself if you want to ask a question quick, or I'll fire one off here. Kennan said something about time of the year. How time sensitive are you to soil sampling right now? Is there an optimum time to be pulling samples, or?
That's I think, a very good question. We tend to think right before planting and right after planting or right after harvest are a good time for the kind of bulk soil sampling. It will change quite a bit throughout the year, but those are kind of good snapshots of what you're looking like going right into growing season and what you're looking at kind of going into that fallow period or going into cover cropping or whatever's going on in the winter. And especially if we do look at both of those, then we can kind of get a sense of, because obviously your nutrients are going to be a bit depleted throughout the growing season by the time you get to harvest, and how the microbiome is responding to that going into winter. Josh, do you have any thoughts on sample timing?
Based off of, we're still looking into the literature. We haven't done any studies where we look at, and this is really helpful for us because right now, this year we collected the soil in the spring, and we're running these tests on your soil and your seed from spring conditions, um, which I think it may be more indicative of what the end of the season might look like. Um, because you've had the chance, the soil's had the chance to rest. Um, but at the end of the season, we're going to get a better snapshot at what sort of pathogens might be in your system and how those pathogens might potentially influence those communities. As it relates to connecting the dots between biological potential of your soil to produce had to have a positive response for the plant. I don't know. That's a great question, Lauren. Something that we intend to do more research into. My hunch is that if you're my hunch is that the closest that we can get to before planting, that's kind of would be, I would say, the ideal sample collection time. But data is suggesting that these microbial communities, the microbes that are in the soil, they'll stay in the soil, they'll survive in the soil. You'll maintain some of the same levels of diversity. It's just that some of them are going dormant in certain times of the year, or but from my conversations with Doug, we feel as if these soil communities they stay somewhat stable throughout the year, but there are slight fluctuations between harvest and spring.
Hey guys, question on the reports right now. We're scaling things as a percentage, 0 to 100. I'm assuming the reference data base for that scale, you haven't found the, perfect soil yet for 100, but you have that in theory. That's evolving, I'm assuming. So my question becomes, as a producer, if I have a sample I sent to you this year, I have a sample I sent to you next year after you have more data into that model, or take the same soil over two different times, does my percentage relatively change? Can I compare samples in my field between years and I see like a, 2 or 5% variation? How much of that is changes in my field versus how much of that is changes in the reference data set, that it's being scaled against, I guess. So just something I'd appreciate some background
Yeah,
on.
that's a great question. And you're exactly right. Those percentiles that you're seeing is based off of a reference cohort of samples that we've grown in the greenhouse up to V2, or just 2 weeks post germination. That is kind of that's really where our technology shines, is being able to baseline rhizosphere communities at 2 weeks post emergence. We've based off of the literature and scientific evidence, we've determined that that is the most predictive growth stage to look at future crop health. And the beautiful thing about metagenomics in all of this data, when we sequence that DNA, it's forward compatible. So as a company, we're discovering new biomarkers associated with nematodes. We're constantly updating our rice score to be more and more and more accurate to reflect what the potential of that root is to produce a positive influence or performance on the field. And we can actually go back in time to a previous report that was generated for you, and update it with our most up-to-date metrics, or re-update on a year-to-year basis as it relates to those communities. Those are things that we absolutely could do, and as we continue to grow and we introduce our platform, which we can talk about a little bit here today. You'll have your own account where you can go back in time and look at previous samples that were collected, so you get that longitudinal data set, and you can understand, and the data becomes more and more and more accurate over time. And so that is an important component in this data, is to have the ability to rerun it through our bioinformatics pipeline to make it most up to date. That's a really good question, though. Thank you for that.
Add to that a little bit, because you're absolutely right that we are constantly updating our reference population. So you will see some slight differences year to year. But like Josh mentioned, we can and will rerun previous year's samples, and so you'll have that comparability. So I know there's some farmers who don't have a sample from last year, but Joe, I know you do. So we'll make sure and deliver, I think, results on both for this year, just so you can. I know you're very interested in those year-on-year comparisons. And yeah, it will change slightly year to year, but we update them kind of real time as we go. Just like Josh mentioned, we have forward compatibility, so we can keep rerunning those over time. And I do see that Jacob had a question as well, and this kind of gets at this project, I think, is a little bit of a one-off for us. The normal cycle for our reports is if you just send us a soil or a root, we try to get those back in around 21 days, about 3 weeks. And if it's a greenhouse grow, if you just send us soil and seed and we're trying to do that greenhouse grow, we usually shoot for about a month. But this was a little bit of a kind of a separate research project where we're growing, I think, 50 different soils in the greenhouse under some different conditions. And so this had a little bit different timeline. So I apologize that we, haven't delivered any kind of insights yet. But if under the normal process, yeah, we would be trying to get those back within 3 weeks or a month. But this is a little bit of a one-off. So just wanted to address that real quick.
Yeah, I think for reference there, Jacob, Josh, if you remember that when you pulled the samples, we seen something specific, and he was trying to track that. And so in the future, that would be a 21-day turnaround then.
Yeah, that's right now our process for getting a sample and sending out a report. Whenever we sequence the material, so once we get access to the rhizosphere, we harvest the rhizosphere from the greenhouse or from the grow box. We get it into the lab, we extract the DNA. It's about 2 weeks to be able to get a report out. And so, and it takes about 2 weeks to grow it out. So we're talking about, it's about a 4-week turnaround. And this is something that we're really excited about. We haven't really socialized, so we're happy to kind of throw it out to y'all and get y'all's thoughts on this. But we are working on kind of a grow box, and it's essentially it's a germination kit. So you go out in your field, collect your soil, you bring it to the grow box, put your soil in the grow box, you plant your seed in the grow box, and then you collect the rhizosphere. We give you the SOPs and the protocols for treating the soil and how to plant them properly, how to harvest them, place them in a bag, send them to the lab, and then we can tell you A/B testing which seed performed optimally based on the Rhize score, which biological input performed optimally versus the Rhize score. And so that is more A/B testing, and it gives you the ability to basically, I like to call it a little mini field trial grow box. Instead of running these large commercial trials out in your field, spending $10,000 on a tote that covers 1,000 acres for a product. Instead of running those trials, we want to try and get it into a small contained environment that's standardized that we've trained our models on to look at those rhizospheres and provide insight into whether or not a seed is going to perform for your specific soil. Wanted to throw that out there. Is that something that y'all would be interested in? Um, um, is it something that you might utilize on the farm?
Anybody have input on that one? Feel free to unmute yourself. I think you stunned them, Josh.
Yeah. Maybe I think uh, a picture might be better. I don't have any pictures of our work there, but yeah, we're excited. We're excited about the opportunity to get those out. I think getting them in the hands um, of agronomists and farmers is going to be important. So, do you think it might make sense? I guess I pulled up this report, our new, I guess, updated report. So this is kind of a sneak peek at what the what the updated report would look like right now. Historically, over the last two years, we've been delivering reports through Excel. And so this is kind of a mock-up of what future reports would look like. Um, Not just providing the bacterial and fungal genera, but also providing a little descriptor of what those microbes are known to do. And then going a little bit further into the pathogen risk, we screen for over I believe it's 600, 700 pathogens for around 60 different crops. So having that pathogen risk assessment tied to the report that we think is going to be important. And then a soil health overview, key findings and considerations for um, maybe ways to improve the metrics that you have on your report. So this is our updated new improved report. And when we go into these one-on-one meetings, we'd love y'all's feedback as to ways that maybe we might be able to improve it.
Josh, I got a question for you.
Yes, sir.
First, I haven't seen my report. I'm hoping you don't tell me you recommend CRP or pasture ground on my farm ground. But question for you is, we've done a lot of soil testing up here, and the variability from taking that soil test, even if you have a GPS location and you go back to that same spot multiple times a year, from a fertilizer standpoint, the variability is pretty massive. There's so many outside criteria that go into that, where how much moisture is in the soil profile at the time of the sample, what time of year is it, where are we at in the frost thaw cycle? Is it right after it thaws, or is it right after we get done with harvest has a major variability on it too? Have you done any testing going back to the same exact location, multiple samples throughout the year to see the variability? So based on that sample, you can kind of feel where that's going to be.
That's a good question. I think we are looking at that data now. So even I would say beyond that, the some of the studies that we're doing now is looking at the rhizosphere, that root zone, at different growth stages of the crop. And so kind of comparing how the root system is developing over time is an important part of the R&D and development that we're working on. But yes, that's something that, I think would be really valuable, is to have that baseline after post-harvest or pre-planting, and having a comparative diagnostic to see what those major shifts or changes might look like. We should have that data on one or two, maybe three farms, and so it might be valuable for us to go through and look at that. But as of right now, I can't really, I'm not fully certain if I could give a good answer there. That's a really good question, and I would imagine that yes, that is the case, that those communities are shifting. Um, in an ideal world, we would do, 5-acre grids, 15 to 20 cores per block, and we'd be sampling in the spring and in the fall. But that can be very cost preventative just because of the cost. And so our theory is that if we can establish a good zone, or representative zone of a field, or a representative of a zone of a soil type that's very common in your field, and we get a good mixture of that soil, we could inform on maybe how that system is performing. And I guess one question for y'all might be, when y'all are buying these inputs, or seeds, how many acres are those seeds going on? Is it is one seed that you're purchasing representing, 200, 300 acres? Is that a common practice? If so, then I think that, if we could get a representative soil type on where that seed is going, we might be able to inform on whether or not there's going to be a positive response. I guess maybe an example would be a biological input. When y'all are purchasing biological inputs, how many acres are those being applied to?
It seems like most packaging is in 40, 80, or larger acre unit packs anyway, is what I'm seeing. 40 is probably the smallest.
Kennan, do you have any insight on that?
No, I would say you're bringing up a good question though, because like our soil types are highly variable. I mean, we've got so many different per field, and when we've been sampling for RhizeBio, we've never paid attention to the soil type yet. So how relevant is soil type to these samples?
I would say it's definitely relevant. That was one of the things, or at least when we were doing sampling down in Tennessee, we tried to stay somewhat within a single soil type, or if you have a field that kind of is split pretty clean into, get a good representation of both. Because like Josh mentioned, ideally we'd love to get, kind of really down in the weeds and be able to sample each and every little portion of the field to kind of capture those nutrient variations, those soil variations. But it's a bit cost prohibitive. So for now, we've kind of taken the approach of, let's get a good composite as best as we can, represent everything in the field to understand kind of across the board how it's looking. I think one of the ways that we try to look a little bit more specifically is through rhizospheres, because the plant will kind of respond to those changes in the local soil environment, and we do take a few more of those. But it's a very good kind of point in question that that variability is a challenge that we fight, I guess I would say.
Yeah.
In any soil testing is a challenge you're going to fight. Like you said, even making nutrient recommendations, you're going to have a little bit of variability based on where you take the sample or the soil type that you took the sample in.
Yeah, and that's a really good point, Keenan. We started out looking at bulk soil DNA and doing DNA sampling just on that soil. And what we realized is that there's a lot of noise. The majority of those microbes are dead or dormant, maybe not interacting directly with the crop. And so when you're looking at a soil sample with this DNA analysis, it's not very informative. It might be informative from a communal health standpoint of saying, hey, we have the balanced fungal to bacterial ratio or mycorrhizal population. But when looking at these different nutrient cycling pathways like nitrogen, phosphorus, potassium, calcium, it's not as informative because you have that other layer of the seed genetics. And so when you place that soil or you collect that soil, if the microbes are there that we're looking at, we get a good homogenized sample. Some of those microbes, they might be dormant, but we're bringing it into a greenhouse, a grow hut, and we are waking those microbes up, getting them exposure to a root sample. And that crop we've seen is the crop is very selective about who it wants from your field because it's engineering and telling and trying to understand who's there, who's in the environment, and that gives us another level of precision that is much more accurate than a bulk soil sample. So to your point, if there are major changes or shifts in active microbial communities from the fall to the spring, there's also microbes they're not going to die off. They're either going to go dormant because the plant doesn't need them or isn't requiring them, but they're still going to be in your system. And so if we can get it into the greenhouse, if we could get access to get those microbes into a system where they can repopulate and find a host, that's really where our technology shines. And our hope is that we can eliminate a lot of that variability because we are able to recruit those microbes that are in your field into a sample that we can inform on what's happening in that system. And really, I guess what we're looking for is symbiosis, right? What is the likelihood of that soil to marry well and partner well with your seed? Is there a strong relationship there? If there is a strong relationship there, then we would see a lot of these nutrient cycling pathways improved. Or plant stress adaptation biomarkers improved. If if the that's one question that we can answer with this technology. The other question is a little bit um, it's a little bit beyond or further from that in the sense that these biological products or even these chemistry pop-up programs that we're implementing, how does that change those communities, and is it favoring microbial populations that are positive for your farm? And so when we add that second layer of discovery into these trials on the farm, it can be very informative as it relates to what is a good marriage of a soil and seed, and what is a good marriage of a soil, seed, and product.
Any other questions, or we going to keep moving forward?
I'm gonna pull up kind of I guess another sneak peek. This is kind of going maybe a little bit into the future of what we're hoping to release sometime in the next 6 to 12 months into the future. So if y'all are looking to implement this type of testing next year, this is something that we can produce, or we're looking to produce. But so this concept of the rise score is, as Kennan had mentioned, it's the ability for your soil and your seed from the rhizosphere to be able to withstand stress. It could be a heat wave. It could be pathogen pressure. It could be all sorts of things that would affect your yield. And so we're tying any sort of I don't want to say recommendations, but any sort of insights as it relates to health to this score. And so understanding on a per soil type basis what that rise score is as it relates to, potential biological matches or potential seed matches. These are things that we're working towards as we continue to improve our ability to look at these systems. Yeah. So for instance, this going back to that historical data, Joe, that you were mentioning, tracking that rise score over time to look at, how you're improving it, looking at some of these detected pathogens that might have a negative influence on the field, and then also what are ways that maybe we can improve that score. Looking at more carbon sequestration, there are products that we've analyzed that have the ability to improve carbon sequestration or stress adaptation or nitrogen cycling. And so trialing those and seeing if we can improve those pathways ultimately is what we can help to support. Kennan, do you have any other insights or discoveries or things that we're learning from these trials conducted this year?
Appreciate the update, guys. One thing going forward, I'd love to get to the point where I could test seed before I order it. The problem is getting the seed from the companies to test and then having a bank of relative soil that's enough, representation to be appropriate for a given field, like Lauren stated, with all the variability in some of mine. Because we're making seed decisions now, or the push is there. I mean, supposedly you can still switch them, but even do I switch from Pioneer to Whiffles or something of that nature, which I'm considering to do, but I've got no background data other than field trials by these companies, or maybe if I'm lucky and independent. I've got an agronomist right now that correlates every single soil type, seed that was planted, whether it was corn on soybean ground, and has that in a spreadsheet anyway she's able to share with me, so I can filter by soil type, by seed, if that's one that's been recommended, and see how that's done across a data set. But it's only within her clientele, which is very broad across state. But yeah, getting more of that information in advance, because it seems like a lot of this we're not necessarily, we're testing or we're finding out either through actual yield after the fact.
Yeah.
And getting more predictive in that is the real economic, key to unlock.
Yeah. Yeah, and that's something that we've struggled with because we have to get access to this rhizosphere before planting. We have to get access to the seed before planting. And so our feeling is, and with discussions with agronomists and seed dealers, nurseries, and things of that nature, um, partnering with your local agronomists, us sending over one of these grow boxes, equipping them with the tools to be able to get access to that root is going to be important. And so, if you have any key partners in your area that have relationships with the dealers, the seed dealers that can break bags, and get some seed bank in order, that's where we can definitely work with those key partners to be able to support those decisions. So um, if you have someone in mind that might have access to a seed bank or has access to the seed dealer, that I think would be a good, wonderful place for a wonderful reference for us to maybe help to distribute these grow boxes into your local region or area. But that's a good point, Joe. And I think something that we've struggled with, but it seems to me as if the agronomists, they have the ability to get access to that seed. I'm not sure if would you say that that's the case for the folks that are working in your area?
Josh, I think, some years you can get seed ahead of time, and other years the seed is showing up when you're trying to plant it. So,
Yeah,
the reliability of getting the seed year over year over year, last couple of years has been fine, but you go back a couple of years before that and it was a problem. So,
yeah. What are your thoughts, Sam, on getting access to the seed, I guess, in the spring, and then conducting these grow hut tests, throughout the year before your next year's decision? Is that something that you'd be open to?
I mean, we're still trying to get comfortable with just understanding how the tests and things work and how we can apply it. And you guys running the tests, so jumping into running the tests ourselves and trying to understand it is somewhat of a stretch, I think. But
Yeah, yeah. Well, working through maybe a local agronomist and to establish those grow huts is, I think, maybe something that we're looking for key distribution partners that have the ability, they have the time to be able to conduct these experiments. We also have a seed bank here as well. So, but the key is, a lot of these different seed types in your area, they're specific to your area. So expanding our seed bank internally, I think, is going to be important as we continue to conduct this testing.
Yeah, I think that would be critical to your retail partners.
Yeah.
You're going to have that trusted agronomist that knows and understands the whole system and can help the farmer understand. I go back to one of our calls last year. He's like, "This is great information, but I have no idea what you're talking about." And I could see a
Yeah.
few eyes glossing over probably when you start talking about some of this. So, what's the go-to-market plan on that? I guess. Are you working with potential retailers or?
Yeah, yeah. We just, I would say, in the last 2 months, we're taking pre-orders right now for these grow boxes. We've got a lot of interest, and it's been primarily on the retail agronomy side because they have the ability to get access to those seeds. And so, um, um, yeah, that ultimately is our go-to-market strategy, is to get in with folks that have those connections with the dealers and everything like that to to do this testing pre-planting.
Okay, we're coming up top of the hour. Any more thoughts, ideals? Or did you want to talk about the future a little bit more, or what? We'll finish this one off and when do you want to start setting up one-on-ones? That might be a question. And
We should, yeah, no, we should be ready. Kennan, I'll throw that over to you. We should be ready in, I would say, this week or next week to have these reports completed, if I'm not mistaken, and getting that data out to y'all.
Yeah,
We
I
should,
think
yeah.
maybe after the Thanksgiving kind of holiday and starting in December would be a good time to try and get people on the horn for that. I think next week is probably a little crazy, people running around, but yeah, maybe that first week of December might be a good time.
First week of December.
one quick question and a reminder. I know we talked about yield data and stuff like that. Are you seeing anything corresponding with yield this year? And then I need to remind the guys that we do need some yield data, if
Yeah,
possible. You
yeah,
have trials.
forgot to touch on that. I would say going into these one-on-one meetings, making sure to have that yield data available, and we can send. It might be helpful if we send before the meeting just kind of the location of the field where the sample was collected, so that you guys have the longitude, latitude information prior to the meeting. I think that would be really helpful.
Yeah, having not seen the yield data, I can't speak on that yet, but definitely excited to try and draw some of those conclusions this year.
Yeah. We have the RIZE score, so we're gonna be referencing the RIZE score and seeing how, I guess, how close we are from an ability to predict out what that specific area of the field yielded. So we're excited to go through that. We felt as if last year we were pretty close, um, and gosh, we had a pretty strong correlation score as it relates to that RISE score and its ability to indicate positive or negative yield based off of one field versus another. And so revisiting that, I think, is going to be important for us again this year.
Any closing thoughts, suggestions, or great having you guys on.
Yeah.
Thinking of that, I do have soil action lined up for the first Tuesday in December. I've got Continuum Ag, and we're going to talk R&D tax credits. The Friday Coffee Club in December. So, kind of got things lined up here.
All
If you
right.
got closing questions for Josh or Kennan, feel free to fire away quick, or we'll end this one here. And thank you guys for jumping on. Like I said, start reaching out when we want to do these follow-up calls. And hopefully some of our guys will, I think pretty much everybody's finishing up here. We should have some yield data on that.
Well, thank you so much, Lauren, for facilitating, and we appreciate all y'all for participating in this year's trial. We're learning a lot, and so we're very thankful and excited about what's next.